Good morning everyone. In this video I'll be talking about various techniques invented during ancient period and the golden era of microbiology. What are the techniques? Okay. What are the different methods? So this is also another topic of Unit 1 of UG SEM1 core paper syllabus of microbiology. So this is Unit 1, another topic, and other topics are already uploaded in my channel. I have made videos on that. You can go and check. So what are the different type of techniques? Okay, techniques is basically number one, that is obviously invention of microscope. Who invented microscope? Antony van Leeuwenhoek. I have already made video on that scientist. So this is a technique, basis of the microbiology. Microscope. Without a microscope you cannot check any kind of a microbes. So this is a technique. Microscope invention they are making, it's a technique. So it's a technique. Number two, that is invention of germ theory of disease. Who invented that? Louis Pasteur, famous Louis Pasteur, who disproves the spontaneous generation and invented the germ theory of disease. Then Louis Pasteur again invented some vaccination techniques. Okay, vaccination techniques and all. Then fermentation. Okay, then pasteurization. Okay, so these all are techniques. What are the techniques? It's not like very big things, but the Unit 1 of microbiology CCF core paper 1 is almost linked with each other, all the topics. If you read about the scientists and their contributions in microbiology, automatically you will learn about the techniques of the golden period of microbiology. Okay, so these are linked. Then there are other techniques like Koch postulate. Why Koch postulate? Because it tells about a disease causing specific microorganism, how they will cause disease. There are 4 criterias. I have already mentioned in my channel, made the videos on that. So this is another technique, another very important technique of Robert Koch, that is isolation of microbes, specifically disease causing bacteria, according to the Koch postulate. But this isolation technique, along with usage of agar to solidify the broth media, like nutrient broth media. If you want to pour it into a plate and make a semi-solid agar, semi-solid solid media, then you have to add agar with nutrient broth. So this is a technique to isolate pure culture of bacteria or any other microbes. You need a semi-solid agar media. That is why agar invention or the usage of agar by Robert Koch is also a technique of the golden era of microbiology. So this is the techniques. Then there are another techniques by Joseph Lister. Remember, I have already told about him in my previous videos and uploaded in my channel. Joseph Lister, who invented antiseptic concept by using carbolic acid during the surgery to sterilize the instruments, surgical instruments, and healing the wound processes to reduce the mortality rate after any kind of a surgery. So Joseph Lister is basically antiseptic surgery inventor. So these are also techniques. And after that, by Alexander Fleming, invention of penicillin plus lysozyme, which are basically used for killing of pathogenic bacteria. Pathogenic bacteria means the bacteria which will cause disease. Okay, so this is also a technique. Invention of antimicrobial agents. This is also a technique. So these are the techniques of golden era of microbiology, which included the contributions of eminent scientists. So if you learn about the contribution of eminent scientists, you automatically learn about the different techniques of microbiology. Okay, so this is basically the microbiology. What is microbiology? What are the techniques? What are the basics of microbiology? What are the basic need of microbiology? Basic instruments of microbiology? So this topic is all about that, and hope you like this video. If you like, please subscribe my channel and share with your friends. Thank you so much. Bye.